Welcome students to Module 4, Plant Growth Management and Pest Control. In this module, we'll explore how plants grow, how they defend themselves, and how we can sustainably manage pests and diseases using scientific tools. Our goal is to understand the balance plants must maintain between growth and defense and how Integrated Pest Management, or IPM, supports both plant health and sustainability. In this module, we will answer the question: how do plants grow, defend themselves, and how do we manage pests sustainably in agriculture? An essential question that we will answer through 3 lessons. This module connects plant biology with real-world farming challenges through the lens of sustainability. We'll be breaking down the module into 3 lessons. Lesson A: how a plant grows and fights. Here we'll explore photosynthesis as the energy engine for plants, and how plants have to make trade-offs, whether to grow or to defend themselves. We'll also learn about plant immune signals like jasmonic acid and salicylic acid, which trigger defenses against different kinds of pests and pathogens. Lesson B: IPM as a system. This lesson focuses on Integrated Pest Management, or IPM. Students will learn the 4 tiers: cultural, mechanical or physical, biological, and chemical methods, and how these strategies connect to circular economy principles by reducing waste and relying on natural cycles. Lesson C: Fast diagnostics. This lesson looks at how technology helps farmers catch plant problems early. We'll compare older lab-based methods, such as agar plates and PCR, with newer field-ready tools. Students will learn how early detection can save money and resources, and how timely information reduces waste and increases sustainability. So, the big idea is this: plants don't just grow, they interact with pests, pathogens, and people. And by studying these systems, we can build smarter, more sustainable solutions for agriculture. In Lesson A, we will start by asking the essential question: how do plants balance the need to grow with the need to defend themselves against pests and pathogens? In this lesson, we will be exploring the processes and steps that plants take to grow and survive, as well as how they defend themselves from pests and diseases. Every decision to produce a leaf or defend against a threat involves a trade-off. Understanding how plants manage this balance will help us better understand why IPM and early diagnostics are crucial in farming today. Before we dive into growth and defense, let's do a quick review of how plants actually fuel themselves. Plants have special structures called chloroplasts. Inside these chloroplasts, sunlight powers a set of reactions that convert light energy into chemical energy that the plant can use. This is called photosynthesis. Here's the basic process. Light hits the chloroplasts, kicking off what we call the light reactions. These reactions produce ATP and NADPH, the plant's short-term energy carriers. At the same time, water molecules are split apart, releasing oxygen, or O2, into the air. Later, that energy, ATP and NADPH, helps build sugars like CH2O, which store energy for growth, defense, and everything else the plant does. So here's a question to think about. If a plant doesn't have enough light or water, what part of this process gets interrupted, and how might that affect its growth or defenses? Let's pause here briefly to discuss. Now that we've reviewed how plants capture light energy, let's look at how they turn that energy into food. Inside the chloroplast, plants run a process called the Calvin cycle. This is the stage of photosynthesis where carbon dioxide, or CO2 from the air, is fixed into sugars. Here's how it works. The enzyme rubisco pulls in CO2 and starts the process of carbon fixation. Step by step, the plant builds small 3-carbon molecules called triose P. From there, those molecules are rearranged and combined into glucose and other sugars, represented here as CH2O. These sugars are the building blocks for everything the plant does: growing stems, making seeds, defending against pests, or storing energy for later. Think about it this way. The Calvin cycle is like the plant's kitchen, using ingredients, that is, CO2, ATP, and NADPH, to cook up the sugars that fuel life. A quick question for you. Why do you think it's so important that plants make sugars instead of only relying on ATP and NADPH from the light reactions? Let's pause here briefly to discuss. In plant biology, sources are tissues that produce more sugars than they use, such as mature leaves which conduct active photosynthesis, and sinks are tissues that need sugars but can't make enough themselves, such as roots, young leaves, fruit, and seeds. Sugars move from source to sink through the plant's vascular transport system. If you change the balance of sources and sinks, say by pruning or fruit thinning, you change how resources are allocated. This concept will come up later when we talk about stress and defense, because plants often reassign their energy depending on what's most important for survival at that moment. Plants go through 5 distinct phases in their life cycle. They start as dormant seeds, then germinate when conditions are right. Next comes vegetative growth with leaves and stems, followed by reproductive maturation with flowers and fruits. Finally, they enter senescence, when metabolism slows and the plant dies. Each stage has different energy demands and vulnerabilities. For example, seedlings are highly sensitive to pests, while flowering plants might be more focused on attracting pollinators. Recognizing these phases helps farmers and gardeners time interventions like irrigation, fertilization, or pest control when they'll have the biggest positive impact. Plants don't just grow randomly. They use chemical messengers called phytohormones or plant hormones to control when and how they grow. Think of phytohormones like text messages inside the plant. One part of the plant sends a signal, and another part receives the signal and response. These signals help plants coordinate major life events. Here are a few key examples. Gibberellins are important for seed germination and also help with fruit development and flowering. Cytokinins promote cell division, which is crucial for overall growth and maturity. Auxins help regulate flowering, direct root and shoot growth, and even help fruits develop. So whenever you see a seed sprout, a fruit ripen, or a flower bloom, you're watching plant hormones at work. Here's a question for you. If you were a farmer, how might controlling plant hormones help you grow crops more effectively, maybe faster, bigger, or at the right season? Let's pause here briefly to discuss. This slide goes a bit deeper into how specific plant hormones regulate growth and development. Gibberellins help seeds break down stored food to fuel germination and encourage the sprouting of leaves. They also play a role in flowering and fruit development. Auxins are key for cell growth and elongation. They help shoots grow upward, influence the development of terminal buds, and also support fruit formation. Cytokinins promote cell division, which is important for overall growth and maturity. And they also interact with other hormones to influence flowering. So, together, these phytohormones act like a control network, deciding when seeds sprout, when stems grow taller, when fruits develop, and when flowers bloom. Now think about a plant you've seen in your garden or backyard. Which hormone do you think is most active at each stage? Germination? Leaf growth? Flowering? Or fruiting? How might a farmer or gardener use this knowledge to improve crop production? Let's pause here briefly to discuss. This slide focuses on cytokinins and how they influence plant growth and development. During germination, cytokinins stimulate cell growth and differentiation, helping structures like chloroplasts develop in new leaves. They also mobilize nutrients, making sure energy and materials get to the parts of the plant that need them most, that is, from source to sink. Another important role of cytokinins is postponing leaf aging, keeping leaves healthy and productive for longer. Remember, cytokinins work alongside gibberellins and auxins to regulate the plant's entire life cycle, from germination to growth to flowering and fruiting. Imagine you're a farmer growing leafy greens. How could manipulating cytokinin levels help your plants stay fresh and productive longer? Let's pause here briefly to discuss. Plants constantly make decisions about where to send their energy and resources. This is called source-sink dynamics. Basically, which parts of the plant are sources of sugar, and which parts are sinks that use or store sugar. Normally, the sugars a plant makes in its leaves flow to the parts that need them most, like fruits, roots, or new leaves. We call the leaves the sources, and those growing parts the sinks. But when a plant experiences drought stress, things change. The hormone abscisic acid, or ABA, increases. This signals that the plant is low on water. ABA causes stomata to close, reducing water loss. The plant redirects sugars to the roots, making them stronger sinks, so the roots can grow deeper and search for water. So essentially, the plant shifts its priorities to survival, supporting root growth rather than fruit or leaf growth. Think about it. If you were a farmer experiencing a dry season, how could understanding source-sink shifts help you decide when and how much to water your crops? Let's pause here briefly to discuss. Source-sink dynamics aren't only affected by stress. They can also be actively managed by gardeners or farmers. For example, when you prune fruit trees, you're removing some sinks, like extra fruits or shoots. With fewer sinks competing for sugars, the plant redirects its energy to the remaining fruits. The result? The remaining fruits grow larger and sweeter. Compare a normal tree with many fruits to a pruned tree with fewer but bigger fruits. If you were growing tomatoes in your backyard, How could you use source sink management to produce sweeter, larger fruits? Let's pause here briefly and discuss. Plants are constantly balancing growth and defense. When resources are abundant, energy goes to growth, building biomass and producing seeds. But when pests attack, hormones trigger a defense mode. Energy shifts toward making toxins, strengthening cell walls, or sending chemical signals to attract allies, such as predatory insects. This balance is influenced by the environment, the type of pest, and the plant's own genetics. Farmers can tip the balance in the plant's favor through smart management practices, such as integrated pest management, reducing the need for chemical pesticides. Plants cannot flee. They deter pests with toxins, thick cuticles, and spines. Producing defenses costs energy, so plants juggle growth versus protection. Plants defend themselves in two main ways: chemical and physical tactics. Chemical defenses include toxins, bitter compounds, and volatile organic compounds, otherwise known as VOCs, that signal other organisms. Physical defenses include thick, waxy cuticles, sharp spines, or tough fibers that make tissues harder to eat. The trade-off is that producing defenses takes energy away from plant growth and reproduction. In farming, understanding a crop's natural defenses helps us decide when to intervene with extra protection and when the plant can handle things on its own. Three major plant hormones help coordinate how a plant defends itself when something goes wrong. Gismonic acid, we call it JA, kicks in when a chewing insect takes a bite. It tells the plant to make proteins that mess with the insect's digestion, and it can even trigger the release of VOCs, those chemical signals that call in predators to help. Salicylic acid, or SA, plays a different role. It's activated when pathogens like viruses or fungi attack. Think of it as boosting the plant's immune-style defenses, helping cells shore up their walls and slow the spread of infection. Then there's ethylene, a general stress hormone. Whenever the plant faces multiple threats at once, maybe heat stress and a pathogen, or an insect in drought, ethylene helps coordinate the whole response so the plant doesn't waste energy reacting in the wrong way. Together, these hormones act like an internal alarm system. They tell the plant where the problem is and what kind of defense to launch. And here's the important part for agriculture. When farmers understand these signals, they can choose or breed crop varieties that react faster and stronger, making plants more resilient without relying only on chemicals. Volatile organic compounds, usually shortened to VOCs, are special airborne chemicals that plants release whenever they're damaged. You can think of them almost like plant perfumes, except these scents have a job to do. When a caterpillar chews on a leaf, the plant releases a specific blend of VOCs that drift through the air. Those scents can actually attract helpful predators, like parasitic wasps, that come in and attack the insects causing the damage. But VOCs don't stop there. They also act as warning messages to nearby plants. When neighboring plants smell those VOCs, they start activating their own defenses before the pest even reaches them. It's a little like hearing a fire alarm down the hall. You get ready before the fire gets anywhere near you. This strategy is brilliant because it's indirect. Instead of spending a ton of energy making extra toxins or building thick, costly defenses, the plant calls in allies and alerts its community. In other words, the plant isn't fighting alone. It's calling for backup. Let's look at a really fascinating real-world example investigated by UC Riverside entomologist Dr. Carrie Mogg that shows just how powerful and sometimes how sneaky plant chemical signals can be. Under normal conditions, when a plant is stressed or damaged, it releases VOCs, those airborne chemical signals we talked about earlier. These scents can warn pests away, or they can attract helpful insects, like parasitic wasps, that come in and protect the plant. It's a pretty smart system. But here's where things get wild. A plant virus called cucumber mosaic virus, or CMV, has figured out how to hack this communication network. When a plant gets infected with CMV, the virus actually changes the plant's smell. Instead of smelling stressed or unhealthy, the infected plant starts giving off the scent of a healthy, juicy, high-quality plant, even though inside, it's not healthy at all. Why does this matter? Because aphids, the tiny insects that spread the virus, are strongly attracted to that healthy plant smell. So they fly in, land on the infected plant, and start feeding. While they're drinking the plant's sap, they accidentally pick up the virus. Then the aphids do what aphids always do. They move on quickly to another plant, but now they're carrying CMV with them. And just like that, they spread the virus to the next plant, and the next one, and the next one. The funny part is that the original infected plant isn't even good food for the aphids. Its quality is terrible. But the virus doesn't care about feeding the aphids. It just wants to spread. And this clever scent trick helps it move through a field much faster. This example shows just how much chemistry shapes the relationships between plants and insects. Plants use scents to communicate and defend themselves. But viruses and pests can sometimes turn those same signals against them in surprising and sophisticated ways. Up to this point, we've looked at how plants defend themselves chemically, like VOCs that call in nearby allies, and physically, with traits like thorns or a waxy cuticle. Now let's zoom inside the plant and ask, how does it actually afford all of this? Where does the energy come from? And what has to be sacrificed when the plant is under attack? Now, in this group activity, you will design a circular loop that shows how a plant makes energy, grows, responds to attack, and then defends itself. In your loop, include one clear strategy of sugar reallocation under stress, a visual showing how source-sink relationships shift, and a brief explanation of the hormonal triggers you think are involved. Things like jasmonic acid, salicylic acid, or ethylene. Add at least one specific defense structure or response, such as VOC release, tougher cell walls, thorns, or leaf shedding. Finally, jot down notes on trade-offs. If the plant redirects more sugar and energy into defense, what does it give up? Growth. Reproduction? Storage? Now we'll move into a deeper, group-based challenge using metabolic simulations. In your groups, your goal is to identify the most effective way to stop a pest while keeping the plant healthy. You will be using web-based digital cell puzzles developed by the Lawrence Hall of Science at UC Berkeley for this activity. First, choose a strategy. This could involve boosting the plant's own defenses, targeting the pathogen directly, or a combination of both. Think about the mechanisms involved and why your strategy makes sense. Next, follow the directions of the game in your chosen strategy, and afterwards, carefully interpret the simulation data. Look for patterns in how resources are allocated within the plant and how the pathogen responds. Based on this, propose a biocontrol target, a specific point where intervention will have the biggest impact. Finally, analyze the trade-offs of your approach. What might the plant sacrifice in growth or reproduction? What risks could your intervention pose to the broader crop system? Discuss how these decisions could be applied in real-world agriculture, considering environmental, economic, and practical constraints. We have covered a lot in this lesson. Let's finish it up by reviewing some key questions. First, how do plants shift their sugar allocation between growth and defense when exposed to drought or pruning? Second, what is the difference between the jasmonic acid and salicylic acid pathways in terms of what types of pests or pathogens they defend against? And third, create a labeled sketch or short explanation showing a mini loop of how a tomato plant responds metabolically to heat stress. Think about how these processes influence yield, quality, and the need for pest control. Take some time to formulate your answer, and then we will share out. These review points connect plant physiology with real-world crop management. Let's go over the review answers. During drought, sugars move toward the roots to support water uptake. After pruning, sugars are redirected to the remaining fruits or shoots, which helps them grow larger and stronger. Jasmonic acid defends against chewing insects by activating defense compounds and volatile signals, while salicylic acid targets pathogens like viruses and fungi, boosting resistance throughout the plant. When plants experience heat stress, stomata close, energy is redirected to protect tissues, heat shock proteins are produced, and sugars are sent to the roots and leaves to support defense. Continuing to Lesson B, IPM as a system, we can come to analyze a core question for modern agriculture. How can we maximize crop yield and quality and minimize pesticide use? Integrated Pest Management, or IPM, offers an answer. It combines prevention, monitoring, biological control, and careful chemical use. The result: fewer chemicals in our food and ecosystems, lower long-term costs for farmers, and healthier crops. IPM is a system, not a single tool, and it works best when it's tailored to the crop and local conditions. IPM is a sustainable, science-based approach to pest control. Instead of relying on one method, like spraying chemicals, it layers multiple pest management strategies. These include cultural practices, mechanical or physical controls, biological control using natural enemies, and chemical control only when necessary. The goal is long-term pest suppression, with minimal environmental harm. It's like having a diverse toolbox so you can choose the right tool or combination of tools for each job, instead of using a hammer for everything. The IPM pyramid organizes strategies from most to least pest or disease preventive. At the base are cultural practices, things you do before pests appear, like crop rotation or planting resistant varieties. Above that are mechanical or physical controls, then biological controls like predators or beneficial microbes. At the top is chemical control, used as a last resort when all other strategies have been unsuccessful at effective pest management. This IPM pyramid reminds us to start with prevention and work upward only as needed. Preventive cultural practices focus on changing how and what we grow so that pest problems are avoided before they can even start. For example, rotating crops helps break pest life cycles, while choosing resistant varieties allows plants to naturally fight off certain diseases or insects. Timing also plays an important role. Planting at the right moment can help crops avoid peak pest seasons. Good sanitation, such as removing crop residues and keeping weeds under control, further reduces pest habitats in the farm. Altogether, these practices create an environment that's far less welcoming to pests from the very beginning. Mechanical and physical controls are direct, hands-on ways to reduce pests. Traps like beer cups for slugs or sticky cards for flying insects physically remove pests. Barriers like row covers prevent insects from laying eggs on plants. These methods are especially useful when pest populations are still small and manageable without the use of chemicals. These control methods can also protect beneficial insects by keeping harmful ones away without affecting the rest of the ecosystem. Biological control is all about teaming up with living organisms to keep pests in check. Instead of reaching for chemicals, farmers can use predators like ladybugs, parasitoids like certain tiny wasps, or even beneficial microbes such as Bacillus thuringiensis, which infects only specific pests and leaves everything else alone. There are two main ways IPM uses biological control. First is augmentation, which simply means adding more of these helpful organisms when you need extra support, kind of like calling in reinforcements. Second is conservation, which means protecting the natural enemies that are already there. Farmers might plant flowers to give them habitat or avoid harsh chemicals that would accidentally kill them. What makes biological control so powerful is how targeted it can be. Instead of wiping out everything in sight, these natural enemies focus on the problem pest. That's why biological control is such an important tool in sustainable farming. It lets nature do a lot of the work, while keeping ecosystems healthier in the long run. In citrus groves, predators like lady beetles, lacewings, spiders, and murids can reduce Asian citrus psyllid, or ACP populations, by 80 to 100% in developing colonies. ACP is a pest that causes significant damage and loss in citrus plants. These predators also help control other pests like aphids and mites. By relying on natural enemies, farmers can keep pest levels below economic thresholds without frequent chemical sprays. This is a real-world example of how biological control supports both crop health and environmental sustainability. The economic threshold is the pest population level at which the cost of crop damage or loss equals the cost of pest control. If pest damage would cost more than controlling them, it makes sense to act. If not, you can save money and avoid unnecessary treatments that would be fiscally counterproductive. For example, if damage costs are estimated at $120 per acre and spray costs are $40 per acre, it's worth spraying the crop. But if damage costs are $38 per acre and spray costs are $40, you should skip it. This approach prevents waste and keeps farming economically efficient. Chemical control in IPM is used only when the other methods aren't enough to control the pest. The focus is on judicious use, which is applying pesticides based on monitoring data, not on a fixed schedule or rate. Selective pesticides target harmful pests while sparing beneficial ones. Resistance management is also key, by rotating chemicals or mixing IPM methods so pests don't evolve resistance. Chemicals are a tool, but they're most effective when integrated with the other tiers of IPM. Many modern pesticides are systemic, meaning they travel inside the plant's tissues after the surface level spraying. This offers protection to leaves, stems, and roots from within, often starting immediately. Systemic pesticides can reduce the need for frequent spraying, but they come with risks. These chemicals can persist in soil and water, affecting pollinators, beneficial insects, soil microbiome, and even human health long after harvest. Pesticides can also gradually accumulate in the environment, thereby leading to ecosystem disruptions. This is why sustainable pest management looks for alternatives or uses chemicals only when absolutely necessary. IPM reduces chemical use, which minimizes pollution and protects non-target species like pollinators. It can lower long-term costs and improve yields by keeping pest populations in check without damaging beneficial organisms, thereby promoting agricultural resilience. However, IPM can take more time to set up and requires careful monitoring. It also depends on farmers having good knowledge of pests and their natural enemies. Sometimes, natural enemies can become pests themselves if not managed properly. IPM is a powerful approach, but success depends on knowledge, observation, and balancing the benefits with practical challenges. Even though IPM has clear benefits, adoption rates can be slow. Barriers include lack of ecological knowledge, misconceptions about IPM's effectiveness, and insufficient farmer training. Cultural constraints, like older farmers who are used to chemicals, tend to be skeptical about newer, sustainable management strategies, thinking them to be less effective. Weak pesticide regulations and misaligned policy incentives can also play a role. Economic constraints, like the cost of biological controls, and the need for better local tools are other challenges. Overcoming these barriers requires education, strong legislative support, and innovation. We've covered how complex agriculture is, discussing the inherent challenges of how plants must balance their growth versus defense, and how we, as growers and scientists, have to balance effective pest management against long-term sustainability. So, what is the path forward for integrated pest management? How do we take IPM and make it the mainstream practice? Future improvements in IPM rely on a few key areas that go beyond the lab. First, we need farmer-centered education. Training programs that are local, practical, and focused on the specific crops and pests farmers actually deal with will give them the confidence and skills to use IPM successfully. Policy changes, like subsidies or incentives for IPM adoption, can help. Cross-sector collaboration is also crucial to share knowledge between researchers and farmers. And finally, building markets for sustainably grown produce creates consumer demand, which is a powerful incentive. By combining these efforts, education, policy, collaboration, and market demand, IPM can become the mainstream approach, making agriculture both productive and sustainable. Now, let's look at the future of research listed at the bottom of the slide. In the past, finding the best balance in a plant was slow, done through decades of field trials. But today, technologies like metabolic models and omics-guided simulations are helping us design sustainable strategies before we even set foot in the field. This is just one powerful example of the new tools scientists are developing to push the limits of IPM by using advanced computational science to engineer resilience. In the next few slides, we will take a moment to look at exactly what a metabolic model is, because it directly ties into your upcoming hands-on activity, the Metabolic Simulation IPM Sprint. We just mentioned how metabolic models and omics-guided simulations are a key future direction for agricultural research. Instead of finding the best balance for a plant through decades of slow field trials, we can now design sustainable strategies before we even step onto the farm. This is how we use science to engineer resilience. So, what is this powerful new tool? A metabolic model is essentially a detailed, digital map of a plant or a microbe's entire life. It helps us understand exactly how living systems process nutrients and allocate energy. Think of it like a complete, digital blueprint for the cell's factory. Every living organism runs on thousands of interconnected chemical reactions, which we call metabolic pathways. Metabolic models map out every single one of these known pathways, since they are built using the organism's complete genome sequence, which provides the instruction manual for every reaction. We often call them genome scale models, or GSMs. They take the blueprint and turn it into a dynamic simulation. There are three core concepts the model helps us quantify. First, the pathways, the sequence of reactions themselves. Second, fluxes. This is the scientific term for the rate or speed at which molecules are flowing through those pathways. Think of it as measuring the traffic flow on that cellular roadmap. And third, trade-offs. Since a plant has a finite amount of energy, if it increases the flux down the defense pathway, the model immediately shows the cost, less energy left for the growth pathway. Modeling is essential for quantifying that trade-off, which is the central theme of this module. The real power of the model is that it functions as a what-if machine. Because the system is digital, we can run millions of omics-guided simulations much faster than in a greenhouse. We can virtually change an enzymatic step or boost a defense pathway and immediately see the precise result. If we increase resistance, does the yield drop too much? This process allows scientists to quickly test and optimize solutions digitally, saving incredible amounts of time and resources before they commit to an expensive, multi-year field experiment. Finally, we connect this cell-level science back to the farm. Genome scale models, or GSMs, are what allow us to perform these omics-guided simulations to predict the precise cost of different strategies. The model's prediction, that is, the modeled change in defense levels, disease risk, or product output, translates directly into a practical field strategy. For instance, the model helps us define an IPM action threshold. It tells us, if you use this nutrient amendment to boost defense, you can tolerate 5% more pathogen presence before you must intervene. This is how we take knowledge of a single pathway and turn it into a sustainable decision for the farmer. This is exactly what you will be simulating in your upcoming metabolic simulation IPM sprint activity. Based on what we've covered so far, here are some questions for you all to reflect on about the real-world implementation of IPM strategies. First, take a moment to reflect on the people who are actually out in the fields every day. How do you think education and training in IPM practices might affect the health and safety of farm workers? If workers learn to scout for pests, use thresholds, and choose lower risk tactics first, how would that impact their daily exposure to chemicals, their protective gear, and their sense of agency on the job? Next, think about the crops and the environment. When a farm really commits to IPM, that is, rotating tactics, using biological control, spot spraying instead of blanket treatments, how might that change the levels of pesticide residues on the food we eat and in the soil, water, and surrounding ecosystems? Finally, here's your call to action. Imagine you're part of a team trying to spread best practices in IPM. How would you share clear, evidence-based guidelines so they actually reach farmers, farm workers, and communities? Would you use social media, workshops, bilingual flyers, on-farm demos, school programs? And who would you partner with? Extension agents, farm worker advocates, local health clinics, grower associations, student groups? Jot down some concrete ideas about both the message and the messengers. On this slide, we are focusing on a real-world example of biological control in an IPM program and how molecular tools help us find the good guys in the soil. The James Borman Lab at UC Riverside studies beneficial soil microbes that protect plant roots. One of their key organisms is Dactylella oviparasitica, a fungus that literally traps and kills root parasitic nematodes. By attacking the nematodes in the soil, this fungus helps keep roots healthier and reduces the need for chemical nematicides. In the image on the right, you can see those fungal loops capturing nematodes under the microscope. That's biological control happening at a microscopic scale. Molecular tools like DNA sequencing allow researchers to accurately identify these beneficial microbes and track which strains are most effective in the field. This is a good example of how biological control can be very targeted and environmentally friendly. It also shows how using beneficial microbes fits into the IPM pyramid as a preventive strategy, taking discoveries from the lab and turning them into practical tools that farmers can use to manage pests more sustainably. Now let's look at another kind of biological control, this time above ground. Lady beetles are insect predators that feed on aphids, giving us a natural way to reduce pest pressure on crops. When aphid populations are high, lady beetles can move in, feed aggressively, and quickly cut down the number of pests. But there's a catch. Their impact is often short-term. Once aphid numbers crash, there may not be enough food to sustain the lady beetle population, so their effectiveness can decline unless we actively support their habitat or provide alternative prey. One strategy is to use banker plants. These are plants that host small, controlled populations of prey insects, so predators like lady beetles always have a food source and can stick around even when pest levels in the main crop are low. Compared to Dactylella oviparasitica, which can establish in the soil and offer longer-term suppression of root parasitic nematodes, lady beetles act more like rapid response agents. Very effective in the short term, but often needing ongoing support to stay in the system. To wrap up, let's compare these two major types of biological control and how they fit into integrated pest management. Microbial biocontrol, like the fungus Dactylella oviparasitica, usually acts in the soil or on plant surfaces. It tends to work more slowly, but once established, it can provide continuous, long-term suppression of pests. In contrast, insect biocontrol, like lady beetles feeding on aphids, acts quickly on above-ground pests. These predators can rapidly knock down pest populations, but they often need ongoing support through habitat management, banker plants, or periodic releases to stay effective. So, microbes are more like a slow, steady defense system in the soil and on roots, while insects are fast-acting responders against pests such as aphids, caterpillars, and whiteflies on leaves and shoots. In practice, farmers can combine both strategies to match their crop, their pest problems, and their management goals. To close, think about this. Given a specific crop and pest scenario, which method would you choose? Microbial biocontrol, insect biocontrol, or a combination of both? And why? Let's pause here briefly to discuss, and then we'll share out. As an optional extension, let's look at a powerful, real world example of metabolic modeling in action. A UCR led project focused on designing drought tolerant specialty crops. Climate change is causing increased drought, which is predicted to cause major yield losses, up to 40% in specialty crops like California citrus. This project is a proactive strategy to adapt the crops themselves to survive with less water. Instead of just reacting to the drought, they are trying to engineer resilience, improving long term sustainability and food security. Here is the strategy they use, which combines lab work with computational modeling. Data collection. They use omics data. Our NESeq tells them which genes are on or off, and metabolomics measures the molecules being produced. This shows them precisely which pathways activate under drought conditions. Build GSM. All this data feeds into the genome scale model to map how water stress reshapes the plant's entire biochemistry. Design solutions. The model points to the metabolic levers they can pull. Short-term solutions involve identifying targeted nutrient amendments. Long-term goals include guiding the breeding or future engineering work for permanently efficient, drought-tolerant varieties. This work connects the high-tech modeling back to our core module themes. For those of you engaging in this optional extended learning, here are 3 questions to drive your discussion. Omics to action. Why is omics data more useful than just measuring a plant's height or yield when building a metabolic model for drought tolerance? Short versus long term. Which solution, nutrient amendments or new varieties, is better for a grower needing immediate drought relief, and which is better for long-term food security? Explain your choice. Modeling the trade-off. If the metabolic model predicts a successful strategy to boost a plant's water efficiency, what is the most likely trade-off you would expect to see in the plant's other key functions, like growth or yield? Let's pause here briefly to reflect and discuss. Now let's zoom out from metabolic models and look at how plants can help build better soil communities between crop cycles. Cover crops are plants that farmers grow not to harvest, but to heal and improve the soil in the off-season. They add organic matter, protect the soil surface, and create a welcoming environment for beneficial microbes. Different types of cover crops bring different strengths. Legumes, like clover or veg, can fix nitrogen from the atmosphere and add it to the soil, boosting fertility for the next crop. Grasses, such as ryegrass, help protect soil structure, reduce erosion, and keep nutrients from washing away. Some support beneficial microbes, such as trichoderma, which help suppress soil-borne pathogens. Together, these plants support a rich microbial community in the root zone, that is, the rhizosphere, which in turn helps future crops grow stronger and healthier. So cover crops are like a living investment in soil health and microbial diversity between cash crops. Can anyone think of a reason why adding nitrogen or protecting soil structure with these plants might be especially important for the next crop in the rotation? Let's pause here briefly to reflect and discuss. For this next activity, you're going to take everything we've talked about and actually design an integrated pest management plan for a real crop. You'll be working in groups, choosing a crop from the UC IPM website, and filling out an IPM pyramid worksheet for that crop. Your job is to pick one tactic at each tier of the IPM pyramid: cultural, physical or mechanical, biological, and chemical control, and then choose one appropriate monitoring method for your pest and crop. Use the UCIPM guidelines as your main resource. They'll give you examples of tactics and monitoring tools that are already recommended for California crops. As you work, talk through your decisions with your group. Why is this cultural practice a good first line of defense? How does your physical or mechanical tactic reduce pest pressure? Which natural enemies or biological controls are realistic for this system? And if you do include a chemical control, how can you use it as a last resort in a way that still fits the IPM mindset? When we come back together, a few groups will be asked to share their IPM plan. How did you decide on one tactic per IPM tier? And why did you choose your particular monitoring method? How might those choices help manage pests effectively while reducing overall chemical use? Let's walk through a concrete example of setting up an IPM plan for greenhouse tomatoes. This slide shows how the IPM pyramid framework works in practice, with cultural, biological, physical or mechanical, and chemical tactics all connected and guided by monitoring. At the cultural tier, We start by planting a resistant tomato cultivar. That choice alone can lower susceptibility to key pests and reduce the need for more intensive interventions later on. At the biological tier, we add releases of Incarcia formosa wasps, which parasitize whiteflies and help keep their populations in check. On the physical or mechanical side, we use yellow sticky traps. These serve two roles. They physically capture some whiteflies and, just as importantly, they act as a monitoring tool so we can track how pest numbers change over time. Chemical control is reserved as a last resort. Here, that might mean applying insecticidal soap, but only if monitoring shows that whitefly populations have crossed an action threshold. So in this example, every tier of the IPM pyramid has a specific tactic, and monitoring ties them together. So chemicals are used only when they're truly needed. Now, let's wrap up this lesson by reviewing some key ideas of what we've covered on IPM. First, what are the four tiers of IPM, and how do they work together to support a circular economy system? Second, how does combining methods improve environmental and economic sustainability of a farm? And third, why is pest monitoring essential in making cost-effective IPM decisions? Take a moment to think about and answer these questions, and afterwards, we will discuss together. Here are the answers to the review questions. The four tiers of IPM are cultural, mechanical, biological, and chemical controls. They work together by preventing, managing, and reducing pests while minimizing waste, thereby promoting a circular economy system. Integrating IPM methods protects ecosystems, prevents pest resistance, and saves farmers money, thereby making their farm operations highly sustainable in the long run. Pest monitoring ensures that interventions happen only when necessary. That is, when the economic threshold has been crossed by the level of pest damage, thereby avoiding unnecessary costs and environmental harm. In Lesson C of this module, we will be answering the essential question: how can early detection of plant diseases improve sustainability and reduce crop losses in modern farming systems? Early detection of plant diseases can save crops, reduce chemical use, and protect food supply. The sooner we identify a problem, the less aggressive the treatment needs to be. Rapid diagnostics allow for quicker, more precise actions, which help farmers maintain yields and reduce waste. Let's look at what actually happens when diagnostics are too slow. In many cropping systems, disease spread is faster than detection. Plant pathogens can move silently through fields long before farmers see any visible symptoms. By the time wilting, spotting, or yellowing shows up, the infection may already be widespread. Think of a lettuce field that can be wiped out by wilt in just a week. If you don't catch that infection early, you're not just losing a few plants. You may be forced to treat entire blocks with more aggressive measures. That means more pesticides, more labor, and more fuel. All of that adds up into higher costs for growers, greater environmental impact, and more chemical residue in the system. The ripple effects go further, affecting farmer income, food availability, and surrounding ecosystems. Fast diagnostics flip that script. Tools like simple paper strip tests or digital disease early warning systems, like Cropin's DEWS, give farmers an early heads up. They can remove or treat infected plants quickly, target only the areas at risk, and avoid blanket sprays. That allows for more precise, proactive decisions that support both economic and environmental sustainability. So, fast diagnostics don't just save money, they save resources, reduce environmental harm, and help protect the food system. Here we see how traditional agar plating works in pathogen diagnostics. Petri dishes with nutrient-rich agar gel support visible microbial colony growth, but only after a day or more. That delay can be problematic in real-world agricultural settings where early detection is crucial. Agar plating also requires sterile lab conditions and trained technicians to avoid contamination. On top of that, not all pathogens can grow on standard agar media, since they may have highly specialized dietary needs which cannot be easily formulated in an agar-based format. Despite these drawbacks, agar plating remains a valuable method in teaching labs, research settings, and some field labs with basic infrastructure. This classical diagnostic technique provides foundational insights into pathogen identification but is simply too slow and resource and labor-intensive for urgent IPM decisions. After looking at agar plating, let's zoom in on two more newer and precise, but still lab-based, diagnostic methods: ELISA and PCR. These tools have transformed plant pathology and research, and they're the backbone of a lot of what we know about plant diseases today. But they're not always practical for farmers who need rapid, on-site decisions. ELISA, or enzyme-linked immunosorbent assay, works by using antibodies to detect specific proteins from a pathogen. When the antibody finds its match, the reaction produces a visible color change, so we know that pathogen is present. PCR, or polymerase chain reaction, takes a different approach. It amplifies tiny amounts of pathogen DNA, making it extremely sensitive and accurate, even when the pathogen is present at very low levels. Compared to agar plating, both can deliver lab results much faster, often within 6 to 48 hours. The trade-off is that ELISA and PCR require specialized equipment, clean lab conditions, specific reagents, and trained personnel. That makes them expensive and logistically challenging for small-scale farmers or remote communities. They're incredibly useful in universities, research labs, and large agribusinesses, but they're not ideal for emergency field-based decision-making in IPM. Now we're shifting from lab-based tools to rapid, field-ready methods. One of the most promising is the paper microfluidic chip, a simple, portable diagnostic tool designed for speed, affordability, and accessibility right where crops are growing. Here's how it works. Wax printed channels are laid on a small piece of paper. When you add a liquid sample, like plant sap or extracted leaf juice, it flows through those channels by capillary action. That means the chip doesn't need electricity, pumps, or any bulky equipment. Inside the paper, reactive chemicals are waiting. If a specific pathogen is present, they trigger a clear signal, such as a color change or fluorescence, that can appear within minutes. These chips have several big advantages. They're fast, they're portable enough to use directly in fields, greenhouses, or smallholder farms, and they're very affordable, often costing less than $1 per test. That makes diagnostics more inclusive, especially for small or remote farming communities that can't easily access advanced labs. Let's now look at two of the most cutting-edge tools in rapid diagnostics: LAMP and CRISPR-Cas12. These methods take the precision of molecular biology and package it into formats that can actually work in farm environments, not just high-end labs. LAMP, or loop-mediated isothermal amplification, copies pathogen DNA at a single, steady temperature, usually around 65 degrees Celsius. Unlike PCR, it doesn't need expensive thermal cyclers. A simple heat block or even a controlled water bath is enough. Because everything happens at one temperature, LAMP can produce results much faster than PCR, often in about 30 minutes. CRISPR-Cas12 goes one step further in specificity. You can think of it like a GPS for genes. The Cas12 enzyme is programmed to search for an exact DNA sequence from a pathogen. When it finds that target, it switches on and starts cutting a special reporter molecule that glows under a small LED light. That glow becomes the readout, yes or no, about the pathogen's presence or absence, respectively. The powerful part is that LAMP and CRISPR steps can be combined in a single tube using portable tools, with the entire test taking under an hour and requiring no major lab equipment. That makes these assays especially useful for small farms or remote areas, and it fits well with sustainable, equitable agriculture. We've now seen a range of rapid diagnostic methods, but what makes a tool truly fast and usable on a farm? It's not just about how quickly you get a result. It's also about smart design, portability, and how different technologies work together in real-world conditions. Let's look at the examples on this slide. First, smartphones. These aren't just for taking photos anymore. With the right attachments and apps, they can detect pathogens right in the field and analyze results on the spot, so farmers don't have to send samples to a lab. Next, lab-on-a-chip systems, like the paper microfluidic chips we discussed, shrink an entire lab test down onto a small, portable device that's easy to carry into fields or greenhouses. And then we have biosensors, which can detect specific targets without needing extra labels or complicated chemistry, making tests simpler, faster, and easier to interpret. So, fast testing isn't just about time, it's about smart, integrated design that actually works in the real world. In the last slide, we talked about the technologies that make testing faster and more portable. Now, let's look at how all those pieces come together in a real device. What actually makes a portable detection tool work in the field? This diagram breaks down the main parts of a modern field-ready diagnostic device. You see the sensor components, things like cameras, GPS, light sensors, microphones, and infrared detectors. These are the eyes and ears of the device. They help it sense its surroundings, whether that's detecting a pathogen on a leaf, monitoring temperature and humidity, or tracking where in the field a sample was collected. Next, we have the actuators, the parts that respond. These include LEDs, screens, speakers, and vibration motors. Once the device has analyzed the signal, these components show the results through light, sound, or vibration, making the test easy to read even in bright sun or noisy field conditions. Finally, we have the applications. These devices can be used for pathogen detection, agricultural and environmental monitoring, and even wearable sensing for field workers. You can think of them like a smartphone crossed with a lab instrument. They can detect, analyze, and report in real time and send data instantly to farmers, advisors, or remote teams. So the key to real field-ready diagnostics is combining smart sensing, clear output, and fast communication, all in a portable form you can actually carry into the field. What if diagnosing plant diseases could be fast, accurate, and automated before the damage is even visible to the eye? This spotlight on Dr. Douglas Hill's work shows how that idea is becoming real in citrus. Dr. Hill and his team at Technology Evolving Solutions have developed an automated system for preparing and testing citrus budwood, commonly used for orchard propagations. Traditionally, prepping and processing these samples is one of the biggest time drains in citrus diagnostics. This tool automates that entire workflow, increasing both speed and precision in detecting pathogens like citrus tristeza virus or Wang Longbing (HLB). By building early detection directly into the propagation process, this system lets growers and nurseries identify infected material much sooner. That means they can isolate or remove infected plants before diseases spread through an orchard or a nursery block. The result is less crop loss, better protection of high-value citrus trees, and a reduced need to rely on heavy chemical interventions. It's a concrete example of how the technologies we've been discussing, sensors, smart tools, and automated processing, come together in a real agricultural setting. What if you could test plant health with just your phone and get results in 15 minutes? This case study from the Tsutsui lab at UC Riverside gives us a glimpse of that future. Dr. Hideaki Tsutsui and his team has developed an ultra-fast, lab-on-a-phone diagnostic test that uses isothermal amplification, a method that works at a constant temperature, so it doesn't need complex PCR machines. The whole system is field portable and smartphone readable. You run the reaction in a simple device, and then use a phone to capture and interpret the result. In about 15 minutes, you know whether a virus is present. That makes it practical not just for researchers, but for farmers and ag technicians working in remote or underserved areas. This kind of tool supports real-time decision-making. Instead of waiting days for lab results, growers can test plants directly in the field, spot infections early, and act quickly to remove or manage infected plants before a virus spreads through a whole block. What's especially exciting is that this platform was originally developed for SARS coronavirus 2 and is now being adapted for plant viruses, showing how innovations in human health can cross over into agriculture. So this is a cutting-edge example of portable diagnostics. Fast, convenient, and built for real-world use in pest and disease management. How might a tool like this change the way a farm handles sudden disease outbreaks? What crops or viruses do you think this would be most useful for? And what challenges might farmers face even if they had the test in hand? This slide answers a simple but important question. Why does speed really matter in plant disease diagnostics? We're going to walk through a quick cost example to see how even a short delay can turn into a big financial loss, and how rapid tests can flip that equation. Imagine a disease moving through a field where unchecked crop loss is about $4,000 per acre per week. If diagnosis is delayed by just 2 days, that's roughly 2 out of 7 days in a week, so about $1,100 per acre in losses. Now compare that to a paper microfluidic chip that costs around $50 per test, but gives you results in about 15 minutes. With that rapid detection, you can catch the problem early, treat sooner, and avoid most of that $1,100 loss. Summed up simply, even if you spend $50 on the test, you save about $1,000 in crops. That's a net gain of roughly $950 per acre. Faster tests mean earlier treatment, less crop loss, less wasted pesticide and fertilizer, and better profit margins overall. So rapid diagnostics aren't just cool gadgets. They're investments that protect food, money, and sustainability. Every hour, every day matters. By investing in smarter, faster diagnostics, we're not just improving science, we're changing the economics of plant health management. Now we'll do an activity that combines everything we've learned about IPM and plant diagnostics. The goal is to identify a crop threat using a rapid diagnostic tool and make smart, timely decisions that reduce waste and crop loss. Start by analyzing a real case scenario. Choose a crop and identify a pest or disease based on symptoms. Then select a diagnostic tool, like LAMP or CRISPR, and justify your choice based on speed, cost, and accuracy. Next, Fill out an IPM plan using one strategy per tier: cultural, mechanical, biological, and chemical, and show where the diagnostic tool fits into the system. Finally, estimate the potential savings and benefits of using rapid diagnostics. Think about how it reduces pesticide use, prevents crop loss, and tightens feedback loops. The goal is to make a circular economy inspired, smarter intervention for the farmer. At the end of the activity, we will have your group share out your diagnostic plan, and we will discuss its implications for the overall IPM plan for that crop. As we wrap up this lesson, it's time to think about what we've learned. First, what are the main differences in speed, accuracy, and practicality between traditional, for example, PCR, auger plates, and rapid, for example, LAMP, CRISPR, diagnostic methods? Second, how might using a rapid field diagnostic tool prevent yield loss in a lettuce crop threatened by fungal blight? And third, where would you integrate a rapid diagnostic tool into an IPM plan, and how does it close the loop on pest control? Take a moment to reflect on these questions and consider how faster, smarter diagnostics can transform crop protection. Afterwards, we will discuss the answers together. Rapid tools like LAMP and CRISPR are much faster and can be deployed directly in the field, while traditional methods like PCR and auger plates are slower, require lab equipment and trained staff, and are more resource intensive. For the lettuce and fungal blight scenario, a rapid field diagnostic test lets you detect the problem early and act immediately. That early detection limits disease spread, protects yield, and cuts down on labor and chemical input costs because you can treat only what's necessary instead of the whole field. Rapid diagnostic tools work best at the monitoring stage in an IPM plan. They close the loop by turning scouting observations into clear, early yes or no answers, so growers can take precise, timely action and avoid unnecessary, broad-spectrum pesticide use. As we wrap up Module 4, there are three main takeaways for you. First, plants are constantly making trade-offs between growth and defense. They only have so much energy, so when pests or stresses show up, they have to shift resources away from growing bigger or making seeds and into protecting themselves. Second, Integrated Pest Management, or IPM, is not a single silver bullet. It's a framework that combines cultural, mechanical, biological, and chemical tactics into a coordinated plan. The power of IPM comes from using the right mix of tools at the right time, not relying on chemicals alone. Third, rapid diagnostic tools change how decisions are made. By giving farmers real-time information about what's happening in their fields, these tests speed up responses, reduce unnecessary pesticide use, and help avoid overreacting when there isn't actually a problem. Together, these three pieces, plant energy allocation, the IPM framework, and better diagnostics, add up to a more resilient and sustainable approach to agriculture. This mind map is our big picture roadmap for the module. It shows how plant growth, plant defense, IPM strategies, and fast diagnostics all fit together in one system, from what's happening inside a single plant to the choices farmers make in the field. In green, you see the plant itself. Plants are always balancing energy between growth and defense. In normal conditions, they grow leaves, roots, and fruit. When pests attack, hormones like jasmonic and salicylic acid switch on and energy shifts into defense. In orange is IPM, our second layer of defense when the plant's own responses aren't enough. That includes cultural tactics like rotation or resistant varieties, mechanical tools like traps or barriers, biological control with natural enemies, and chemicals as a last resort. In blue are diagnostics, tools from auger plates to ELISA, PCR, LAMP, and CRISPR that help us catch problems early. Early detection means less damage, better decisions, and less wasted time and money. Career pathways in the topics we covered in this module can be threefold. Firstly, careers in plant science include plant physiologist, molecular biologist, crop scientist, greenhouse technician, breeder, or botany research assistant. These roles focus on understanding and improving plant growth and health. Secondly, in pest management and sustainable agriculture, careers range from IPM specialist to agricultural extension agent, biological control researcher, farm manager, or sustainability consultant. These jobs blend science and problem-solving skills. Finally, AgriTech and diagnostics careers include plant pathologist, diagnostic lab technician, AgriTech product developer, precision agriculture technician, and agricultural data analyst. These roles focus on applying technology to improve plant health and farm efficiency. In this module, we connected plant biology to pest control strategies and modern diagnostic tools. We saw how science helps us grow more food with fewer resources while protecting the environment. As you reflect, remember our framing question: how do plants grow, defend themselves, and how do we manage pests sustainably in agriculture? Studying plant growth and protection shows why a plant's natural defenses aren't always enough. When pests or disease get through, strategies like integrated pest management and rapid diagnostics are essential to prevent major crop losses. Here's a quick preview of the 3 hands-on activities you'll do in this module. First, you'll run a multi-day IPM mini lab using yeast, apple discs, and a simple balloon assay to test cultural, biological, and chemical control strategies. Then use your data to design a small IPM plan of your own. Next, you'll make DIY spore prints from a powdery mildew-infected leaf onto agar plates, and compare fungus growth on a water control plate versus a clove oil-treated plate, calculating how much a natural product can actually slow a plant disease. Finally, you'll go digital with the Metabolic Simulation IPM Sprint, using the digital cell puzzles available online via the UC Berkeley Lawrence Hall of Science to tweak cell-level processes inside plants or microbes, run virtual experiments, and then translate your best strategy into an IPM pyramid and field action threshold for real citrus systems.